Good afternoon, everybody, and welcome to this afternoon's webinar, the first in a series that we've got planned for 2015. Today's presentation, Why We Use Cell Lines as Models, a 30-year journey and what lies ahead, will be presented by Jim Cooper, a cell biology application specialist at the European Collection of Cell Cultures. Jim has over 20 years' experience of delivering authenticated cell cultures for researchers in academia and industry, and for many years has played a key role in managing internal development projects, custom cell culture products, and ECAC's internationally renowned cell culture training courses. Jim is also a member of the International Cell Line Authentication Committee, also known as ICLAC. Now, before we get started, I'd just like to draw your attention to the question button at the top of the page. Should you have any questions during the presentation, please feel free to type in the questions and we'll do our best to answer them at the end of the discussion. So, Jim, it's over to you. Thank you very much.
Thank you, Ian, and thank you everybody out there for dialing into this webinar. So why we use cell lines as models? A 30-year journey and what lies ahead. The European Collection of Cell Cultures, or ECAC, is part of the Culture Collections of Public Health England, which is a not-for-profit venture operated by the Department for Health, which includes three other biological collections: the National Collection of Pathogenic Fungi, the National Collection of Pathogenic Viruses, and the National Collection of Type Cultures. Together, the four collections hold over 50,000 strains of microbes and cell cultures of clinical importance. Our mission is to ensure the outputs of worldwide life science research are credible and reproducible through the use of authentic microbiological strains and cell lines. 2015 marks the 30th anniversary of the establishment of ECAC. To celebrate, we're going to run a series of webinars focusing on the use of cell lines together with our international distribution partners, sigma-aldrich, with whom we've had a strong working relationship for the last 15 years. So cell lines and cell cultures have been essential enabling factors in research for decades, and it is highly likely that they will continue to do so for many years. This webinar will focus on the advantages, limitations, and history of cell culture, with particular reference to why we use cell lines and the way cell culture and ECAC has evolved over that time. Going back to the middle of the last century, early cell culture techniques were driven by two main disciplines: cancer research and the production of antiviral vaccines. As highlighted in this rather politically incorrect article from the New York Times in 1955 that describes the bulk production of Hela cells for the testing of the efficacy of polio vaccine. However, nowadays we see cell cultures as being used in most areas of life science research. In research into intracellular synthetic and metabolic activity, such as nucleic acid synthesis, translation and transcription and protein production, intracellular flux, processing movements of cellular products, for example, RNA and signal transduction, genomic studies, proteomics, and cell-to-cell interactions. And in the fields of applied biology, cell culture has been key in biotechnology applications, especially since the initial decision to use only primary cultures for the production of human biological products was challenged in the late 1960s by the introduction of human diploid cells, and again in the 1980s by continuous cell lines. Cell culture also enables immunology, pharmacology, tissue engineering and regenerative medicine, and toxicology. As a tool, cell culture has many advantages. We can control the macro and micro environments, the pH, the temperature, osmolality, dissolved gases, hormones and nutrient concentrations. We can regulate the cell matrix, control cell-to-cell interactions, and gaseous diffusion. Using perfusion techniques, we can model in vivo conditions. And through selective culture, we can derive homogeneous, reproducible populations of cells. And through appropriate testing, ensure the purity and identity of our cells, allowing validation and accreditation. We can relatively easily characterize cells using cytology, DNA profiling, and immunostaining. Cells can be cryopreserved and stored in long-term liquid nitrogen storage and recovered with little loss. The use of cell cultures in experiments allows the setup of many replicates and effective statistical analysis. Cell cultures can be grown in low-volume systems, making efficient use of reagents. Experiments can be set up with accurate dose concentration, and samples can be taken at specific time points, allowing accurate dose-response analysis. Cell culture processes lend themselves to automation and miniaturization. Experiments and assays normally generate data in a short time frame, which saves time. And cells are ethically preferable to the alternative, the use of animals. But cell culture also has its limitations. It needs a specific expert skill set to cultivate and maintain pure and valid cell cultures. Scientists need to be experts in aseptic technique, and they should understand the biology of the cells and the system they're working with, including the metabolic and environmental needs of the cells used. And unlike microbes, cells are not usually capable of independent growth, and therefore medium compositions are complex. Self-discipline, tight control of procedures in the facility, and an ability to identify and troubleshoot problems is critical. Scientists should be aware of the issues of cross-contamination, mycoplasma contamination, and overpassaging, and how to avoid, test, and ensure pure, authentic cultures. Finally, cell culture is inherently an inefficient process, producing very little yield of biomass for the expense and effort required, and can be quite costly. In general, in cell culture, our aim is to ultimately establish a differentiated culture that represents the physiological tissue or disease state. The phenotypic properties of the original tissue are rapidly lost in conventional in vitro culture through a process historically termed as dedifferentiation, which in reality is probably the overgrowth of the culture with undifferentiated cells. Under correct conditions, differentiated properties can sometimes be restored, as could be seen here in the induction of A549 lung cancer cells to adopt an alveolar type 2. Apologies, I've flipped over 2 slides then. So if you look here on the left-hand side, you can see A549 cancer cells adopting an alveolar type 2 phenotype where they express multilamellar bodies and surfactants. The majority of conventional continuous human cell lines have been derived from cancers and are subject to genetic instability due to their aneuploid chromosomal makeup. It's demonstrated here on the right in these 2 karyograms, the top showing the normal human karyotype and the lower from a breast tumour displaying abnormal chromosome numbers and translocations. This can cause variability in the cultures from one culture to the next and will be made worse through continual subculture, highlighting the need for controlled cell banks. A significant proportion of the literature, some people estimate 20%, has been carried out on misidentified cells. A comprehensive knowledge and understanding of this issue is required by anyone researching, planning, or performing cell culture-based experiments. This isn't a presentation on cell line identity, but it is critical. This year, the International Cell Line Authentication Committee member, Dr. Christopher Korsch, published his findings on his investigation into the financial and intellectual costs of the cell line misidentification. Christopher calculated the cost of the inappropriate use of 2 misidentified cell lines, HEP2 and INT407, both confirmed as Hela over 40 years ago, yet still cited incorrectly. He found that there were 5,789 articles in over 1,000 journals in which HEP2 was used inappropriately, and over 1,000 articles in 271 journals for INT407, all seemingly escaping the peer review process. Together, these invalid publications generated over 200,000 citations and cost research institutes, grant funding bodies, and industries and taxpayers over $4 billion. Most cell culture is carried out in the majority of laboratories in two-dimensional monolayers using monocultures of a single cell line, whereas in vivo cells exist in three-dimensional structures and interact with other cell types, matrices, and are under the influence of the nervous and endocrine systems and paracrine signals from their surrounding neighbours. As demonstrated by Otto Warburg in his Nobel Prize-winning research in the first half of the 20th century, cancer cells and most in vitro cell cultures exhibit an aberrant metabolism, with most of the energy generation being carried out through the glycolysis in the cytosol, rather than a citric acid cycle. Proliferating cell cultures and cancer cells effectively ferment glucose to produce 85% more lactic acid than normal differentiated tissues, rather than using physiological aerobic respiration. This means that in conventional log-phase culture, cells in vitro are more metabolically similar to tumours or healing wounds than differentiated tissue. It's vitally important that scientists understand the limitations of cell culture. Yet, despite the drawbacks, cell culture still is a powerful and useful tool for life science research. And over the last 30 years, cell culture has helped deliver the in vitro production of tissues for regenerative medicine and toxicology applications, such as skin and neurons, biotherapeutic proteins and monoclonal antibodies, vaccines, gene therapy, animal cloning, an understanding of aging, tumour biology and virus genetics, novel drugs, and even an in vitro cultured beef burger. Before looking back over the last 30 years, it's worth a quick review of some of the significant milestones in cell culture history. In 1907, Ross Harrison published a short paper entitled Observations of the Living Developing Nerve Fibre, where he described his research on the growth and development of the nervous system through studying fragments of tadpole spinal cord, which had been incubated in a clot of lymph in a hollow ground glass slide. A technique not dissimilar to cutting-edge 3D hanging drop technologies available nowadays. Here we can see a schematic of this apparatus and the images and data that Harrison acquired using his camera obscura microscope. Harrison found that nerve fibres grew from the explants by active movements of the nerve fibre tips. And through his work he resolved one of the major anatomical controversies of the time, that the nervous system was made of discrete neurons rather than a continuous reticulum. In 1912, Alex Carrel, a great publicist of cell culture, and his team at the Rockefeller Institute were working on the problems of wound healing and nerve regeneration. Without the aid of antibiotics, Carrel, using his skills and knowledge of asepsis as a surgeon, applied tissue culture methods to these problems using explants of chick heart. Obsessed with maintaining physiological environment, he kept the temperature at 39 degrees centigrade in the lab. He painted the walls dark grey and insisted his technicians wore black robes that covered their faces. Carrel tried to avoid any unwanted deviations from physiological conditions found in the body, and he also introduced the quantitative aspect of cell culture, measuring the diameter of the tissue explants he generated. Years ahead of his time, he established an unlikely working relationship with the aviator Charles Lindbergh, who, inspired by his sister-in-law's mitral valve heart disease, had developed an oxygenating pulsatile pump to keep organ cultures alive. The article on the right, from Modern Mechanics magazine in 1936, speculates that Carrel's techniques could enable a booster heart to engineer and help improve human performance. Yet, despite claiming to keep chicken heart tissue in culture for decades, Carrel's work could not be reproduced. And as was suggested by Amanda Capes Davies last year, this may be the first recorded case of cross-contamination in the literature, with Carrel and his co-workers inadvertently supplying their cultures with cells along with the cell culture medium. From our standpoint in the 21st century, we could easily fall under the misapprehension that the early cell culture techniques of Carrel and Harrison were primitive, but they were highly sophisticated, well controlled, and generated high-quality data, and it demonstrated extraordinary skill. With Earle's isolation of L cells from mouse tissue and their subsequent cloning by capillary technique by Catherine Stanford, The 1940s saw the establishment of the first continuous cell lines. We also think of 3D cell culture as being a novel development, yet as long ago as 1947, Hogue reported the production of organotypic slice cultures that retained the structural integrity of the original tissue. A major breakthrough came in 1949 when John Enders, Thomas Weller and Frederick Robbins cultivated poliovirus in tissue culture using monkey kidney cells. The team was subsequently awarded the Nobel Prize. Their original intention was to cultivate chickenpox virus, but they found the virus growth in their negative controls, and almost by accident they had cultivated poliovirus that could then be taken forward into a vaccine. The inset on the right, taken from the Smithsonian Institute, shows their apparatus: flasks in which poliovirus grew in monkey kidney tissue, and the wooden roller used to rotate tubes containing the growing virus. In the 1950s, 60s and 70s, cell culture was made accessible, easier and standardised, enabling consistent growth of proliferative cultures. Renato Doveco used the enzyme trypsin for the first time to subculture cell lines whilst he was working on cancer-inducing effects of polyomavirus. The use of trypsin made cell culture an accessible technology for many scientists, and on the one hand introduced a level of standardisation and reproducibility, but on the other put a selective pressure on most cell cultures, selecting for cells insensitive to damage by trypsin. In 1952, George Guy announced the world's first continuous human cell line, Hela, from a sample taken from the cervical cancer of the patient Henrietta Lacks. A controversial story and a tale well told in Rebecca Skloot's book, The Immortal Life of Henrietta Lacks. Over the decades, Hela cells have enabled many scientific advances. However, they've also cross-contaminated and invalidated dozens of other cell lines. Historically, glass vessels were used for cell culture. The new generation of polystyrene flasks held a great potential. They could be made sterile, disposable, and they were optically perfect. However, they had one major drawback. Their hydrophobicity made them unsuitable for cell attachment. Before they could be used, they'd have to be coated in collagen. In 1975, another tissue culture advance, Charles Einstein and Paul Hartmann developed the plasma discharge method for treating polystyrene. This heralded a new generation of easy-to-use, sterile, disposable flasks. The advantage of this technology was that cell culture became readily accessible for most scientists. Cell lines were suddenly easy to grow, sterility was easy, and conditions could be standardised and reproduced. But all of these advances drove cells into a proliferative, non-differentiated state, and perhaps fueled the misconception that proliferation is desirable and normal. They also put the same selection pressures on most cell cultures, driving cells into a state where perhaps they have more in common with each other than the original tissue or tumour. It's also worth noting that none of today's tissue culture techniques would be possible without the development in the 1970s of the laminar flow biosafety cabinets that provide a safe and sterile environment for us to handle our cultures. As tissue culture became more standardised, it became important to develop quality control procedures. In the late 50s and early 60s, Louis Coriel and others demonstrated the importance of mycoplasma contamination in cell cultures and detected them by culture isolation. And as long ago as 1967, the issue of Hela cross-contamination was first highlighted by Stanley Gartler, and then confirmed by Walter Nelson Rees a decade later. An issue that is relevant today as it was nearly 50 years ago. During the 60s, 70s and 80s, there were a series of advances in techniques and our understanding of the biology of cells in culture. As part of their research into human ageing, in 1961, Leonard Hayflick and Paul Moorhead defined the finite lifespan of normal human cells and identified the Hayflick limit. Their studies demonstrated that normal human cells are only capable of about 70 population doublings before they came senescent. Serum-free media were an important milestone, as they avoid the issues of variability between batches of serum and the potential for unwanted biological agents in the manufacture of biotherapeutics. And in 1975, Caesar Milstein and George Kohler made an important breakthrough when they first fused B cells from immunized animals with multiple myeloma cells, and with an elegant selective cell culture technique, generated the first monoclonal antibody-producing hybridomas. In 1976, the totipotency of embryonal stem cells was identified, and a series of advances came in 1978. Molecular, cellular, and developmental biology selective serum-free media was developed by Richard Ham, and that enabled selective growth of specific cell types. Judah Folkman and Anna Moscona were able to manipulate the adhesivity of cell culture surfaces to control the extent of cell spreading to produce a series of quantitative cell shapes. They found that cell shape was tightly coupled to DNA synthesis and growth in non-transformed cells, suggesting this mechanism is important in the growth of mammalian cells. Marcellino Tarito established differentiated polarized epithelial cells from using MDCK cells on permeable supports and demonstrated trans-epithelial electrical resistance, or TER. In 1979, Tom Maniatis and his team transfected mammalian cells with prokaryotic genes and maintained expression with selective cell culture conditions, paving the way for gene therapy and a new generation of recombinant cell lines. In 1980, John Hassel was working on proteoglycans to determine their role in disease and development. Hassel isolated a heparin sulfate-containing proteoglycan from the Engelbroth-Holm swarm sarcoma. This reagent is now known as Matrigel. In 1981, Evans and Kaufman reported the establishment of mouse embryonic stem cells. This was a major achievement, and the cells were able to differentiate in vitro, after inoculation into a mouse as a tumour. It took, however, another 17 years for James Thompson in Wisconsin to repeat this achievement with human embryonic stem cells. In 1983, Evans and Tim Hunt identified a protein specified by maternal messenger RNA in sea urchin eggs that was destroyed at each cleavage division. They called this new protein cyclin. Later in the decade, Paul Nurse and Tim Hunt, together with Leland Hartwell, went on to understand how cyclins regulate cyclin-dependent kinases to give us great insight into the checkpoints that control the cell cycle. And they were awarded the Nobel Prize in 2001. The 1980s saw the development of many specialised cell lines and cultures, including keratinocytes, reconstituted skin cultures, human mammary epithelial cells, and human microvascular endothelial cells. And then, in 1984, the National Collection of Animal Cell Cultures was established at Portland Down by Professor Brian Griffiths. And a year later, it was redesignated the European Collection of Animal Cell Cultures, or ECAC, in recognition of being an international patent repository. A few years later, we dropped the animal in the title, but retained the A in ECAC. Originally, with only a handful of staff and a few cell lines, the collection soon established itself as a major international cell culture organisation. ECAC immediately started to play a major role, supporting the emerging field of genomics. In 1986, ECAC was awarded an EU Biomed grant to establish a human genetic cell bank based on a generation of EBV-transformed cell lines from patients and families with rare chromosomal and genetic diseases, preserving their genomes to provide limitless supplies of DNA for research. And between 1990 and 2000, with MRC funding, ECAC was able to play a key role in the delivery of the Human Genome Mapping Project, accurately providing DNA from EBV-transformed authenticated cell lines derived from thousands of patients with known mutations and disease states. Since then, ECAC has supported dozens of genome-wide association studies and other large cohort studies through banking network grants and Wellcome Trust funding, allowing research into nephritis, diabetes, Alzheimer's disease, asthma and eczema, motor neurone disease, heart disease, to name but a few. In 1989, Robert Weinberg from MIT used cell culture techniques to discover the first human oncogene, RAS, and the first tumour suppressor gene, retinoblastoma protein. And with these findings, he went on with Douglas Hanahan in 2000 to define the hallmarks of cancer in their landmark publication in the journal Cell. Concerns over the suboptimal use of cell lines in cancer research, particularly the drawing of conclusions about specific tumour on research of a single or a few cancer cell lines, were partly addressed in the establishment of the Cancer Cell Line Encyclopedia by the Broad Institute. Large cancer cell line panels offer the potential to model the genomic diversity of human cancers. The Cancer Cell Line Encyclopedia is a compilation of gene expression, chromosome copy number, and massively parallel sequence data from 947 human cancer cell lines. Baratina and colleagues coupled the pharmacological profiles for 24 anti-cancer drugs across 479 of these cell lines in a panel, which allowed the identification of genetic lineage and gene expression-based predictors of drug sensitivity. And many of these cancer cell lines are available from ECAC. The requirement for better differentiated physiological models and the needs of regenerative medicine have driven the development of novel technologies for generating scaffolds with 3D cell culture and tissue engineering. Historically, it was believed that cellular differentiation was a one-way process, and terminally differentiated cells could never return to a stem cell-like state. Yet, in 2006, this dogma was overturned, and the textbooks rewritten. 2006 saw the demonstration of induced pluripotent stem cell reprogramming. iPSC, as it's now known, was first demonstrated in mice by Shinya Yamanaka's lab in Kyoto, Japan, and then in humans, simultaneously by Yamanaka and also by James Thompson's lab in Wisconsin. John Gurdon and Shinya Yamanaka were awarded the Nobel Prize in Physiology and Medicine in 2012 for the discovery of iPSC reprogramming. This diagram shows how iPSC can be generated from, for example, adult skin cells. They can then be reprogrammed by the activation of the transcriptional regulators OPT3/4, SOX, KLF4, c-MYC, and NANOG. Then, in theory, these iPSC can be differentiated into any tissue of the body. Not only does this have great potential for regenerative medicine, holding the promise of repairing damaged or diseased tissues or organs with the patient's own cells, avoiding any complications of rejection, the technology can allow the in vitro culture of cells of any desired tissue from patients with specific diseases. iPSC techniques are being rapidly developed. Originally, the transcriptional regulators had to be introduced by viral vectors, but novel virus-free techniques are being developed, such as reprogramming using valproic acid. By way of an example, Gareth Miles at St Andrews University has recently used iPSCs derived from patients' skin samples to derive motor neuron-like cells. Using these cells, Miles and his co-workers have shown that in motor neuron disease patients, even before they show any signs of damage, cells affected by motor neuron disease lose the ability to generate the electrical signals required to make muscles contract due to changes in ion channel proteins. And here, this image on the right shows the differentiation of these cells into neurons, demonstrated by staining for markers beta-3-tubulin, HB9, SMI32, and glial fibrillary acidic protein. With dozens of labs in the UK and Europe attempting to realise the potential of iPSC technology, there are thousands of cell lines from diseased and normal individuals in the pipeline. Last year saw the establishment of the European Bank for Induced Pluripotent Stem Cells, or EBISC. Led by Pfizer and managed by Roslin Cells, this European consortium comprises 8 active participant centres with clinical and patient networks, a global leader in industrial iPSC supply, international experts in iPSC science, biobanking, bioengineering, regenerative medicine and data management, and experts in law and ethics. EBISC will spearhead Europe in the international standardisation of iPSC banking by forging collaborative links with similar endeavours in the USA and in Asia. And at ECAC, we are proud and excited by our status as a consortium partner, and of the fact that ECAC, Public Health England, will coordinate cell line distribution. There are general concerns over the use of cell lines in research. For example, in a recent paper, Nestor and colleagues reported in primary mouse cells a dramatic and novel consequence of adaptation of mammalian cells to culture, which suggested a rapid loss of methyl cytosine dioxygenase activity, meaning that epigenetic and transcriptional reprogramming occurs much earlier than was previously assumed, potentially having significant implications for the use of cell lines as faithful mimics of in vivo epigenetic and physiological processes. Further concerns over reproducibility in life science research were raised by Bigley and Ellis in Nature in 2012. And these could be partly addressed through identity testing of characterising and standardising cell lines and cultures. To help drive standardisation in culture in 2012, ECAC, together with other international cell line collections under the leadership of Amanda Cates Davis, established ICLAC, the International Cell Line Authentication Committee, with the aim of making cell line misidentification more visible and to promote awareness and authentication testing as effective ways to combat it. So what are the next 30 years? Going forward, we will probably see iPSC and trans-differentiated cells being used alongside conventional cell lines. In the field of iPSC, we may move to biochemical rather than genetic and viral reprogramming. Physiologically relevant cell culture techniques and a tissue engineering approach will ensure we have cells that behave in a more in vivo-like state in our experiments. Miniaturization and microfluidics will help maximize the efficiency of cell culture experiments. And through stem cells and 3D cell culture, we may realize some of the potential of regenerative and personalized medicine and further reduce animals in research and toxicology. To achieve physiological models, we need to drive our cells to differentiate towards a physiological phenotype by employing 3D cell culture techniques, filter wells, and selected medium. We can assess the phenotypes of our cultures using technologies that allow us to analyze and quantify phenotypic properties of cell lines, for example, image data. And in many ways, we are turning full circle, echoing the pioneering work of Harrison and Carell from almost a century ago. So where are these technologies taking us? 3D scaffold technologies such as electrospinning offer the potential to generate 3D matrices that mimic the in vivo cellular environment. Another versatile off-the-shelf technology is the quasi-vivo system from Kirkstall. This system offers plug-and-play simplicity and the potential to link modules, each containing cultures grown in different 3D culture environments, with flow. Here, work by Iori and colleagues in Italy shows how tissue glucose and fatty acid metabolism can be modelled with linked quasi-vivo modules containing hepatocytes, endothelial cells, and adipose tissue. Note that the endothelial cells are held in laminar flow chamber, mimicking blood vessels. This study demonstrated the capacity of this three-way cell culture system to maintain or restore normal circulating glucose concentrations in response to insulin, and showed the effects that these conditions had on other metabolites involved in glucose and lipid metabolism. Differentiation can not only be induced by chemical and hormonal means, but also by physical manipulation of the cells and by pulsatile flow. On the left, you can see technology from the mechanobiology laboratory in Pittsburgh that uses mechanical loading or stretching to induce terminal differentiation in tendon progenitor cells. And on the right, a bioreactor with pulsatile flow from 3D Biotech, again echoing the work of Charles Lindbergh and Alex Carrel. Synthetic three-dimensional scaffolds such as Albertex encourage tissue-like growth. Data on the growth of primary hepatocytes and HepG2 cells on Albertex from Stefan Shabursky here demonstrate that a more in vivo phenotype is generated in the 3D cell culture environment. Filter wells, first used in 1978, are a generic, reproducible technology that drives epithelial polarization and offers the potential for the recapitulation of complex in vivo cellular organization. As shown here by Kelly Berube from Cardiff University, bronchial cells that have been grown on filter wells on the centre image generate polarized functional epithelia that recapitulate the in vivo state. And in comparison, you can see histologies from the bronchial biopsy right and cells grown in 2D culture on the left. Here, a pragmatic approach to the use of filter wells is demonstrated. This approach uses the advantages of conventional cell culture, and the filter well technology. Progenitor cells are isolated from the patient and expanded in conventional tea flasks before being plated into filter wells, and subsequently differentiated into cultures that mimic complete epithelia. If you remember, in 1907, Harrison used the hanging drop technique to culture his tadpole neurons. Novel hanging drop plates, in this case perfector plates, can be used to generate 3D spherocultures in multiple replicates. Other available technologies for spherogeneration include Corning's ultra-low attachment surface and the NASA rotating wall vessel bioreactor. The protocol for generating multicellular spheroids is outlined here. This is from work by Cordelia Bartel from the University of Veterinary Medicine in Vienna. Spheroids of epithelial cells are cultivated in a hanging drop. Once established, a central lumen forms due to apoptosis. The spheroid is then transferred to a well and stromal cells are added. The resultant co-culture produces a model of differentiated glandular epithelium. Bartel then goes on to show how this technology can be combined with the filter well to produce complex 3D co-culture models. In this example, smooth muscle cells are grown on the bottom of the well in a collagen matrix. Stromal cells and glandular epithelial cells are cultivated on the surface of the filter well membrane in an extracellular matrix, which is then, in turn, overlaid with a surface epithelial culture. This model putatively demonstrates epithelial and stromal crosstalk with the surface and glandular epithelia. To provide multiple replicates and a high throughput readout, it is important that 3D culture is available in multi-well format. This technology here allows the potential to run multiple replicates and assays on a nanofiber scaffold in random orientated nanofiber microplates. Here they can be seen supporting the growth of stem cells in this scanning electron micrograph. Microfluidics and miniaturization offer the promise of delivering maximal data using minimal cellular materials, removing the need for extensive proliferation to achieve enough that would be normally required to provide enough cellular material for conventional experiments. The Mimitas organ-on-a-chip system offers the ability to grow and analyse perfused co-culture systems. And they also have a confocal data here showing the generation of HUVEC-derived differentiated capillary-like tubes. We saw at the beginning of the presentation that as long ago as 1907, Ross Harrison used his hanging drop method combined with a camera obscura microscope to generate qualitative and quantitative real-time imaging data on his neuronal cultures. At ECAC, we now use automated live cell microscopy in the form of the Essen Incucyte Zoom to capture qualitative imaging and quantitative data on our cell cultures as they grow. The advantage we have over Harrison's equipment is that we can combine conventional high-definition phase contrast imaging with two fluorescent channels in a walkaway system. We can read several multi-well plates or flasks and dozens of cell lines at a time, and all of the data is safely stored automatically for quantitative analysis and compilation as time-lapse movies. Here you can see data from an experiment on red-labelled Hela cells in which apoptosis has been induced using stavsporin. Apoptotic cells are labelled green with the proprietary caspase reagents available from Essen. And here I've used the Incucyte and red-labelled Hela nuclite cells to model Hela contamination of a fibroblast culture, both qualitatively on the left and quantitatively on the right. So in summary, cell lines are not perfect models. The choice of protocol used to grow them or differentiate them can increase their validity. It is important to understand the advantages and limitations of cell cultures. And it's important to know what's the biological question being asked, and what is the most appropriate system to answer that question. It is, of course, important to ask, are my cells authentic? And do I need proliferating or differentiated cells in my experiment? And are my cells in the right condition or stage of differentiation to answer the question being asked? When using cell lines, don't expect them to immediately behave like differentiated tissue straight out of the vial. Expect to have to search or develop the correct differentiation protocol. Tissue culture, in some ways, is going back to its roots, preserving and recapitulating the in vivo phenotype. To achieve this, do I want a bespoke system or a generic off-the-shelf system? And if I use a complex bespoke system, how will others reproduce my work? Learning from the past, the systems that become commonplace are usually those that are accessible, affordable, easy to use, and reproducible. Well, thank you very much for listening. There are some acknowledgements here, particularly Dr. Ian Freshney, who provided, I used his book and some of his publications in compiling this presentation. Professor Stefan Schabowski from the University of Durham, Dr. Amanda Capes-Davis from ICLAC, and my friends and colleagues from the Culture Collections of Public Health England and within sigma-aldrich. Okay, thank you very much. I'll pass back to Ian now, and we'll start to collate in any questions if there are any.
Okay, thank you very much, Jim. Very interesting. Okay, so we do have a number of questions that have come in. So I'll take them in order. Hopefully you'll be able to answer them all. The first one's quite an easy one, but I'll let you answer it anyway. It's so can we purchase ECAC cell lines via Sigma?
Of course you can, yes.
Yeah, I think everything's...
I have to add that ECAC and Public Health England is a not-for-profit organisation, but in order to get global access, have to use Sigma, and Sigma serve us very well as distribution partners.
Cool, thank you. So, the second question that came in was, currently how many cell lines do ECAC have available but don't include any primary cell lines, only the established ones? Do you know that number off the top of your head?
Not exactly, but the current collection of conventional cell lines stands at something like 1,500. It may be slightly more than that, but over half of those are human cell lines in origin, generally derived from human cancers.
Okay, thank you. So somebody else asks, will the use of differentiated stem cells eventually replace the use of cell lines prepared from different tissues?
Well, that's a really good question, and it's one that quite often, well, it doesn't keep me awake at night, but I do think about this. The thing is that we do know about cell lines that are properly authenticated, is we do know that they did come from an original tissue origin. We know that they will have some memory of their original tissue. Stem cell-derived cells, we have to qualify in their differentiation. And I think particularly with iPSC cells, we don't necessarily have the characterization tools readily available to effectively prove that those cell lines are remodeling the tissue that we want to. We get some idea that they're going that particular way, and there may be some key markers that we can detect. So I think it would be great if iPSC and embryonic stem cell derived cultures could in some ways replace cancer cell lines in modelling normal tissues. But I still think because the cancer cell lines are so well so reproducible, and we can always return to validated stock, they will still always have a place alongside the new cell lines as they're developed, particularly effectively as controls, I think.
Okay, thank you. So, next question, they're starting to come in thick and fast now. Do you think that 3D cell culture can be used for vaccine production? And if so, should it promote a better yield of viral particles?
I don't know, because I haven't tried it. So I think this is one for experimental design. I'm sure that what we will see with 3D cell culture is the ability to cultivate strains of viruses that have hitherto proven very difficult to cultivate. And that's really where it will come into its own, is in the cultivation of difficult to cultivate viruses. In terms of yields and biological manufacture, that's really one to be trialled in production. But I would hope that some of the more awkward viruses, such as the hepatitis viruses, will be able to be cultivated in 3D culture systems.
Okay. So, somebody would like to know, if possible, the procedure, or the best procedure, for transferring cells from a monolayer type into suspension form.
Oh yes, okay, so 3D, adaptation to a suspension phenotype.
Mm-hmm.
There's various methods to do this, actually, and a method that we had pioneered here at ECAC by a previous colleague, Dr. Elizabeth Penn. If that person wants to send in their details, we can send them the protocol. But in essence, what happens is you supplement you have a serum-free media that you want to adapt your cells into. Now, generally, when you have cells growing in an attached phenotype, and you adapt them into a serum-free culture, generally those cells will adapt into a suspension phenotype. So the thing to do is to select one or even more potential serum-free media that you wish to adapt the cells into, and it's a process, a long-term process of subculturing your cells into the serum-free media. You mix the serum-free media with the serum-containing media in appropriate quantities, and gradually over subcultures you withdraw the serum and the basal media that you would have the serum in. So let's say, for example, you're going into a proprietary serum-free media and your cells normally grow in D-men with 10% FBS. You'd mix those media one-to-one with each other, and gradually, time, you would withdraw the D-men and the serum to a point where the cells were adapted into the new media, and generally, without serum, the cells will lose their ability to attach.
Okay, cool. I was just thinking, actually, for people who need protocol information and stuff like that, maybe what we could suggest is that they contact their local Sigma rep and actually say that they've, watched the webinar and that they were interested in certain information, and then we can get that forwarded on. So it's probably the best way to do it. So if you contact your local rep, any questions about protocols, if we've got them available, we're happy to share, but we'll get them to the right people. So next question, should we be using primary cell culture rather than cell lines? Is 2D culture a thing of the past? Well,
2D primary cells grow in 2D as well. That's one thing to be aware of, and that paper by Nestor and other people have shown that effectively primary cells soon lose their ability to represent differentiated tissues, so they effectively de-differentiate over passages. Primary cells have great advantage, and you would expect them to have more physiological relevance, and in some ways, of course, they do. The big drawback with primary cells is that they have a limited supply. So you have supply that's restricted to one donor or one animal. Once you've used that material up, then it's gone forever. So unless you're very, very clever with your cell banking procedures, you will eventually lose that material, and you have to go to another donor. And then, of course, individual to individual variability comes into play, and you will get different. You may get different results. So again, this is an example where we should be looking for physiological relevance. We should be going to primary cells where we possibly can, but of course their supply is limited and their reproducibility is limited as well. And that's why very often people set out with the best intent of using primary cells and often fall back onto the reliability of continuous cell lines.
Okay, thank you. So we have some other requests for protocols here as well. So somebody wants to know the best protocol for growing BHK21 cells in suspension. If that person who sent that in, and this is a quick, easy answer, again, like I said, contacts us. We can get you that protocol if it's not already attached to the website. Again, somebody else asked for that monolayer to suspension, and somebody else has asked for the slides for the seminar. So this is being recorded, and so you'll be able to go back and review it as well. So that shouldn't be a problem for those people. One other question. So how do you ensure, I think you touched on it briefly, but how do you ensure that the cells that you've got in the repositories are mycoplasma free?
Okay, so all cell lines that were received and curated here at the culture collections are fully tested for mycoplasma contamination using three methods. So we use the tried and tested method of culture isolation, which is still the most sensitive test, but also because not every test detects every single species, we also test using complementary methods. So we use Herc stain testing and also mycoplasma PCR. There are other, of course, available techniques for testing, and the more techniques you use, the better, because as I've already mentioned, different techniques or different mycoplasmas evade detection by some techniques. So we are very confident that all of our cell lines test negative. Very few of the cell lines in the collection have been eradicated of mycoplasma using mycoplasma remover agents, and those will always be will always stay in our information people should handle those with suspicion and handle them in quarantine. So using what we believe to be the best available tests, and using a panel of those tests, we're pretty confident that our cells are as free of mycoplasma as they can be.
Okay, so we've got about 4 minutes left. So somebody has just quickly dropped we need to know the suitable antibiotics to use with the monolayer. I guess I've just put a message up. I guess that depends on the cell type. Or have you any suggestions for which suitable antibiotics, apart from saying none at all?
Exactly. Well, this is the thing. Of course, from a pure standpoint, and all of the cell culture carried out at the culture collections at ECAC here, all of our cell culture is carried out in antibiotic-free conditions. So we rely on the asepsis and good laboratory practice to keep our cells free of contamination. I'm well aware that people working in shared laboratories, and also anybody working with primary tissues, may have the need to add antibiotics to their cultures. And of course, generally the first line of defence is to use penicillin and streptomycin, so broad range antibiotics. If you do get contaminations in your culture, I'd recommend using those at first, and then weaning your cells off them, so that you don't stand at risk of using antibiotics continuously. Of course, the best strategy for dealing with a contaminated culture is to, if you have clean stocks, is to throw that culture away and go back to your validated and clean cell bank, and resuscitate the cells from that and start again. So in my experience, so if you do have very precious stocks you need to treat with antibiotics, then pen and strep is the first line of defence, and then you move on to something like gentamicin, or polymyxin B, that kind of thing, until neomycin. But always with short, sharp bursts of antibiotics to try and kill the infection, and not risk having the development of resistant clones of the microbe in the culture. But I believe Sigma actually, I think somewhere in all of the literature that you produce a nice table of antibiotics and their effectivity. So I think there may be something in the Sigma back catalogue actually that could help out there.
Okay. I don't recall off the top of my head, unfortunately. Thanks for that, Jim. So one final question. I think I can probably answer this one myself. It's an easy one. Somebody's asked about the third edition of the fundamental techniques in cell culture. Is there a new one, and has it been printed for distribution? So the answer is no, not yet. We're working on that one.
Perhaps if people would let us know what they want in there, that would be quite useful. Because,
yeah. So edition 2 is very popular, but we're working on updating that. So if there is new techniques, or if there are requests for things to be included, we're more than happy to listen. But that's something that it's maybe something that will come out towards the end of the year, hopefully. That's the plan anyway. And I think we have, we have 30 seconds. So is there a quick detection method for mycoplasma contamination?
Yes, there is. It's a technique. There's PCR, and also there is a technique called MycoAlert, which is available from Lonza, which is a luminescence-based technique, which is very quick and quite accurate. Okay,
cool. And that gives us 5 seconds, so I'm just going to say thank you very much, Jim. I hope that was of interest to everybody. If you need any further information, please feel free to contact us. And as I mentioned, this has been recorded, so you can go back and review it as well. So with that, thank you very much, Jim. I'm going to end the webcast. Thank you very much.